My name is Anshu Kumar, and my topic for presentation is nephelometer and turbidity meter. These are the analytical devices used to measure the turbidity of a solution or liquid. So, turbidity meter is a device used to measure the turbidity or cloudiness of a liquid. So, turbidity refers to the suspended particles in the liquid. So the suspended particle does not let the light to pass through the sample uh, through a straight. So the visibility meter measures the intensity of light scattered or transmitted. Based on that gives the turbidity of solution. So the unit used for the representation of turbidity is NTU, and that is nephelometric turbidity meters. So indirectly, the turbidity meter measure the concentration of suspended particle present in the solution. So in the photo we can clearly see that the turbidity meter in the left side. On the top of the turbidity meter, we will place the samples here. We will place the samples, and we can see the result. That is turbidity of a solution or the sample in the screen. And on the right side we can see the samples of different turbidity. That is, we have a sample of 600 NTU, 50 NTU, 16 NTU, and zero point 0.3 NTU. We can clearly see that for 600 NTU turbidity sample, we cannot we cannot see through the sample, while for the zero point 0.3 turbidity NTU, zero point 0.3 NTU turbidity sample, we can clearly see through the sample. This is the concentration of suspended particle in zero point 0.3 NTU turbidity sample is very low, while in the 600 600 NTU sample, the concentration of suspended particle is high. Now we will see the working principle of a turbidity meter. So the working principle of turbidity meter is based on the scattering and absorption of light by suspended particle. So let's see the working diagram of the turbidity meter or nephelometer. These are both are the analytical device used to measure the turbidity of any sample, as we have discussed earlier. The basic difference between both of them is that the detector used in nephelometer is at 90 degree to the sample, while while in the turbidity meter the detector is at 180 degree. 180 degree. So in nephelometer, we are measuring the scattered light, while in the turbidity meter we are measuring the transmitted light. So when the concentration of a sample is low, we are using nephelometer, while when the concentration of a concentration while the concentration of a sample is high, we are using the turbidity meter. So we can clearly see that the instrument consists in turbidity meter is a light source, a lens, and a sample holder and a detector. The light source in turbidity meter uses a stable light source, typically a LED, laser, or tungsten lamp. The wavelength of the light source is selected based on the standard of for turbidity measurement. Light is directed through the sample, and as it encounters the suspended particle, it scatters in various directions. So, as light enters the sample particle within the liquid, scatter the light in all directions. The extent of scattering depends upon the particle size, concentration, shape, and color. The larger the number and the size of the particle, the more scattering occurs, resulting in the higher turbidity. So the scattering of light occurs by the effect called Tyndall effect. And the longer the wavelength, light is more transmitted, while the shorter the light is more reflected by scattering. So and we have a lens which is used to concentrate the light on the sample. After passing through the sample, we have a detector. A detector, typically a photodiode or photomultiplier tube, is positioned at the specific angle relative to the light source. As we have discussed earlier, in nephelometer, the detector is placed at 90 degree from the incident beam light, while in turbidity meter, the detector is placed at 180 degree. After that, detector measure the intensity of light scattered by the particles. As turbidity increases, the intensity of light scattered light also increases, allowing the instrument to quantify the turbidimeter. For the holding of sample, we have a sample cell, which is shown in the figure. This is the sample cell. So they are in cylindrical shape, flat faces to minimize the reflection and multiply the scattering. So the calculation of turbidity in turbidity methods, the turbidity meter converts the intensity of a scattered light into turbidity reading, commonly expressed in NTU. The meter typically have a microprocessor that interprets the detected light intensity and compare it to the calibration standard and output a numerical value representing the turbidity level. So we can see the in this graph as let's say we can see the graph in voltage versus turbidity meter. So let's say the detector detect the voltage of 3 volt. So from the graph we clearly see that clearly see that the turbidity of that sample is around 800 NTU. So this graph is for turbidity meter. For the nephelometer, this graph is used. As we can see that in this graph, the voltage varies linearly with the NTU, because as the concentration increases or turbidity increases, the concentration of the solution will increase. As the concentration increases, the light will be more scattered, and more light scattered create more voltage. We can see that in this graph. Yeah, so we have previously discussed the nephelometer, where we use nephelometer and turbidity meter. So nephelometer is used when the concentration is low and uniform scattering is happening. Also, the in this device, the detector is at 90 degree, while in the turbidity method used when the concentration is high, scattering is not uniform, and in this the detector is placed at 180 degree. Now we will discuss the importance of turbidity measurements. So the it is important to measure the water quality. High turbidity indicate poor water quality, potential harmful for drinking water and aquatic life. Also it is used for turbidity health purpose. Turbidity can indicate concentration and carrying microorganisms. Now let's see the application of turbidity measurements. So it is used in water treatment, ensure safe drinking water by monitoring salinity. It is used in industrial process for quality control in food and beverage and pharmaceutical industries, etc. And it is also used for environmental monitoring, like for the measuring the turbidity of lake water or river water. Also it is used for the aquaculture, which tracks the water quality for fish and aquatic life health. uh, that's all from my side. Thank you for listening.